started doing the assignment. Have you? Okay. I had given you a date last week. That was 14th September. But since the class was published only on Friday, I am extending the date to 19th. 19th Saturday. Okay, is that clear? So you have to submit the assignment by 19th Saturday. Okay, so please read the chapter, attend the class, collect maximum points and do it in the way I had asked you to do. There won't be any compromise with the neatness of the work. Today I thought of explaining the experiments. That is the first experiment, preparing a temporary mount of onion peel and cheek cell. I had already given you the notes for it. At least you would have read once while writing the notes. Today I will be showing the experiment with the help of a video. You can understand it properly. When we get a chance to come back to the school, we will do it from the lab. Okay. So already I have given you the notes. So while attending the class, please keep your notes near you and see if you have written all the terms correctly in your notebook. I had asked you to write the explanation in the rough record. I told you to take a 100 pages notebook and write it down. So keep that notebook in front of you when you attend this class. And as you attend, certain terms may be new to you. So when you attend the class, please check the spellings and the sentence etc. and do the necessary corrections. After that, I'll give you instruction to do it in the record book. Okay, so that is your work for this week. You have to do all the corrections necessary in your rough record. So after I call the meeting, I will give you all the instructions related to the record book and then you may copy the same into your record book. So first experiment, preparing a temporary mount of onion peel and cheek cell. So when you write the heading itself, you have to write to prepare a temporary mount of the next line A onion peel, next line B cheek cell. Draw the line above and below. So for all the experiments, you will have aim, materials required, procedure, observation, precautions. Inference will also be there for some. So these subheadings should be written neatly with the pen itself and underlining should be done only with the pencil. You should not use color pens or color pencils. Ordinary pen should be used for writing and underlining should be done by using a pencil. Okay, so after write the subheading as a paragraph, you can start and write the remaining things. Then if you want, you can leave a line, write the next subheading, write the things. That is how you have to write neatly in the record. Your left hand side, that is the unruled side, you have to use only pencil. When you open the notebook in front of you, right side you have to write with pen and left side you have to write with pencil. Border should be drawn for the page. Heading should be written with pencil. Underlining should be done with pencil. Drawing should be done neatly in the center of the page with pencil. And labeling should be done on one side or on two sides. By using a scale, you have to draw a line away from the diagram and label it. Never draw the lines with free hand. Don't draw a line in any direction you want. The line should be drawn only towards the sides. If at all it is too congested, you can draw the line upwards, bring it to the side and label it. Hope this instruction is clear. Right side you have to use only pen. Left side you have to use only pencil. Labeling, drawing lines, everything should be done with pencil. Now I will explain the first one, first temporary mount, that is onion peel. When you watch the video, this will be more clear to you. How it is done, what is, when I use certain terms, you may not be knowing. You would have known it because last year also certain experiments I have taken to the lab and shown you. So certain materials may be familiar to you. Certain materials may not be familiar to you. So when you see the video, those things also will be familiar. Okay. If you have got a record book, if the shops are open, if record books are available in the shops, you may buy a record book for your record book. If record books are not available in your locality, if it is difficult for you to buy, then you may use a college notebook for your record book. Till last year we have been using record book itself. This year, because of this lockdown and other things, okay, so certain places are containment zones, shops may not be open, so it may be difficult for you to get a record book. Only because of that, this year we are permitting you to do your record work in the college notebook if record books are not available. You buy an unruled notebook or one side ruled notebook. You may get one side ruled and other side unruled page. So one side ruled notebook, if you get, you buy that. If that is not available, you can buy an unruled notebook for record book. So please don't start at the record. I will tell you, I will give you instruction to start. That is in the Zoom meeting. After your class, I will be recording a Zoom meeting. In that meeting, I will give you instruction related to the record book. So now shall we go to the video?
When you watch this video, please go through the notes which you have already written related to these practicals and do the necessary correction. Sometimes the words may not be written correctly or the spellings may be wrong. So look into this video and correct all the spelling mistakes. Please don't write in the fair record. In the Zoom meeting, I will read this experiment for you and do the remaining corrections. Only then copy it in the fair record. Now let us see how to prepare a stained temporary mount of onion peel. Materials required. First, medium sized onion, slide, cover slip, watch glass, forceps, knife, needle, distilled water, glycerin, dropper, blotting paper, saffron in solution and a compound microscope and a brush. Procedure. First, take a medium sized onion and cut it laterally into 4 to 5 pieces with the help of a knife. Remove a scale leaf from one of the pieces. These are the scale leaves. Green color leaves are called foliage leaves. Break the scale leaf gently from the middle and peel a small portion from the inner side of the leaf that covers the inner surface. You can see the membranous sheet, transparent sheet. Remove the delicate epidermal peel with the help of a forceps and place it in the watch glass containing distilled water. Take another watch glass and place in it 3 to 4 drops of saffron in solution. So it is the stain. With the help of the brush, transfer the peel in water into the saffron in solution for staining. Wait for 2 to 3 minutes. After 2 to 3 minutes, transfer the peel back to the watch glass containing water. This will help you to remove the extra stain sticking to the peel. Take a clean glass slide and pour 1 to 2 drops of glycerin. And then transfer the stained peel on the drops of glycerin on the side with the help of the brush. The peel may tend to curl up because it is membranous. With the help of the needle, please unfold it. Place the cover slip on the slide in such a way that no air bubbles enter into it. Gently press the cover slip with the needle to spread glycerin and peel properly. Remove the extra stain from the slide with the help of a blotting paper. Examine the slide under the compound microscope. Observe the slide first under low power and then under high power. Observation. There are large number of cells lying side by side with distinct cell wall. Cell membrane, nucleus is seen in the center, almost towards the periphery. You can see cytoplasm, and the major part is occupied by the vacuole. So the same diagram you can draw in your notebook. Inference. The cells under observation are the plant cell. It is because the cell wall and large vacuoles are clearly seen. Let us see how to prepare a stained temporary mount of human cheek cells. Materials required: toothpick, cover slip, methylene blue stain, glycerin, glass slide, wash bottle, brush, needle, microscope, blotting paper and a dropper. Procedure. First, take 2 or 3 drops of distilled water and put it on the glass slide. Then, take a toothpick, wash it properly, and before that, please wash your mouth properly. Just scrape the inner side of your cheek gently and put the scrape, that is the cells which you got, on the glass slide on which the distilled water is put. With the help of the needle, just make a smear of it. Okay, just spread it. Then put a drop of methylene blue stain on the glass slide. 1 or 2 drops you can put. After that, we have to put the cover slip. Okay. Before putting the cover slip, if you want, you can add 1 drop of glycerin so that the cells will not dry. Stain is used to see the cells clearly under the microscope, whereas glycerin is put in order to prevent the slide from drying. Put the cover slip carefully by not allowing the air bubbles to enter. After that, with the help of the needle, just press on top so that excess liquid will come out, that is the stain will come out. Then with the help of a blotting paper, just clean the excess fluid that has oozed out. So the slide is ready to be seen under the microscope. Observe the slide first under low power and then under high power of the microscope. It's a compound microscope. When we observe the slide under the microscope, we can see many cells. So next is observation. The cells which are seen, human cells, may be irregular in shape, maybe almost oval or round or rectangular in shape. But we can see that there is no cell wall. Animal cells do not have cell wall. They have only cell membrane. We can see a prominent nucleus in the center of the cell. You can see the darkly stained structure is the nucleus and cytoplasm. Conclusion. Animal cells do not have cell wall. They have only cell membrane. They are irregular in shape because only cell membrane is present. Okay, and there is a prominent nucleus in the center of the cell. Precautions. First, ensure toothpick used to scrape the cheek is clean so as not to cause infection to the cheek. Extra stain should be removed by using blotting paper.